Let's turn our attention now to some basic functional anatomy of the human visual system. We're going to focus in this lecture exclusively on what's often called the primary visual pathway. And the primary visual pathway starts at the eye, so we'll talk a little bit about the eye, the general structures of the eye, how it functions. We'll focus in on rods and cones and their ability, their sensitivity to light and to specific wavelengths of light, how they are distributed throughout the retina. And then we'll turn our attention to ganglion cells. These are the cells that ultimately leave the eye, forming the optic nerve. And we're going to focus on two important aspects of ganglion cells. On the one hand, we're going to note that visual processing is already going on. Even at ganglion cells, we have visual processing that is breaking up that image intensity array at the back of the eye using two kinds of cells, center on and center off cells, that are essentially trying to highlight differences in image intensity to pick out features of the visual scene like edges. In addition to that, what we've discovered in maybe the last 20 years or so really is that there are multiple processing streams in vision, and even in the primary visual pathway. Originally, it was thought that when processing leaves the visual cortex at the back of your head, it goes off in two different processing streams, and that's the point where those streams diverge from one another. But as researchers continued to investigate the nature of the eye, the nature of the primary visual pathway, they discovered that these processing streams actually start all the way back in the retina, that they break off into three distinct streams that are dissociable from each other, though not completely. They interact with each other at various points, and they're named the parvocellular, the magnocellular, and the coniocellular streams. And we'll talk a little bit about the cells that make up each kind of processing stream and the sorts of information they're processing. Then we'll turn our attention to the next big place here on the visual pathway. We'll look at the optic chiasm, the place where our optic nerves from each eye cross over, and representations of the visual array as it's been processed so far from one side of your eye, of both eyes really, go to one side of your head, and the processed information from both sides of your eyes from the other side go to the other side. So there's this crossover in the optic nerve. And that information then goes to the second big stop on the primary visual pathway, the lateral geniculate nucleus. It's a body in the thalamus. It's kind of small, really. But we'll look at the lateral geniculate nucleus and what processing occurs there. And we'll note two important features about the lateral geniculate nucleus. One is that it has a layered structure, and each layer has its own specialized representational role, and that all of these layers are topographically or retinographically mapped. That is, places next to each other in the retina or places next to each other in the visual field are also next to each other on these layers. Now, there are 6 layers in the lateral geniculate nucleus, at least in the areas that are focused on processing information from the fovea, that really small area of highly concentrated cones. As you move out, there are fewer layers and much more sparse processing at the lateral geniculate nucleus, but we'll look at that in some detail. Then we'll move on to the third and final stop in the primary visual pathway, the visual cortex. And we'll look at the different kinds of cells and the sorts of information that they are selectively sensitive to, that they are processing or representing in the visual cortex. And we'll note again, right, that there is a number of different layers in the visual cortex, and that these layers exhibit a topographic mapping as well. So here is our primary visual pathway. It starts with our eye. The eye then transmits its information along the optic nerve. The first major processing stop is the lateral geniculate nucleus here, a body of the thalamus. And then from there, processing streams go back here to the visual or occipital cortex. Our guide for this lecture is going to be Gatto, the kitten with freakishly large eyes, which seems somehow appropriate. So let's take a look at eyes. So our eyes have a cornea. It's a glassy, transparent front covering the eye, and it provides about 2/3 of the refraction of the bending of light into focus from the visual stream. On the outside of our eye, abutting the cornea, is the sclera, the tough white tissue that begins where that cornea ends and creates the envelope that is the eye. Between the cornea and the lens, we have what's called the aqueous humor, the clear water liquid filling that the area has between that cornea and our lens. The iris is that pigmented tissue that can contract and expand, right? So it allows for increased or decreased light, and it's triggered by ambient light. The pupil, of course, is the open area, the area that light travels through, that gets increased or decreased in size as a result of the constriction of the iris. Inside the eye, excuse me, we have a gelatinous, diaphanous fluid filling that eye sac. It's called the vitreous humor. And unlike the aqueous humor, it's colored and thicker. And it provides substance to the body of the eye. The lens is the optic element of the eye that's composed of water-soluble structural proteins. And this part of the eye can be expanded or contracted by these muscles here, the ciliary muscles that support that lens. Of course, as you get older, those ciliary muscles calcify, as does the lens itself, and it becomes harder and harder for those muscles to contract or relax that lens and adjust its refractive properties. Hence, people have a harder and harder time seeing small print as they get older. Those ciliary muscles are connected to the body of the eye, to the suspensory ligaments, the fibrous tissue that connect the lens and the ciliary muscles. The retina is the multi-layered organ that covers about 65% of the surface of the vitreous chamber. So this area around in here, right? It runs roughly from the ciliary muscles to the optic nerve. The fovea, depicted here as this glowing little dot, is 3 to 5 millimeters of retinal area there. It's located slightly off-center towards the temple, and it's that area that contains all those tightly packed cones. The vast majority of the cones, the photosensitive cells that are tuned to specific wavelengths of light that are smaller than the rods, the other photosensitive kinds of cells, packed in very tightly in that foveal area there. And finally, we have the optic nerve, which is the axons from those ganglion cells of the eye. So as the ganglion cells leave the eye, they come together to form the optic nerve. And this creates in here this blind spot, this area where there aren't any photosensitive cells because the optic nerve is occupying this space. And of course, your brain fills in for that blind spot, just creates its projection of what is there in the visual field